Hello students, how are you? I hope that you will be fine and will be busy in your preparations. Today, I, Akash from SciAstra is here to solve 50 important questions of biology with respect to IAT exams, which will ultimately help you in understanding the important sections or important topics of biology, and ultimately it will help you in getting or improving your scores in the IAT exam. Okay, and this session is actually going to help both PCB and PCM students to get or to enhance your marks in IAT actual exam. Okay. So let's begin this session by solving the first question. Okay, let's see. So the first point that I want to make here is that it is not a guarantee that these questions will be asked in the exam, but it is guaranteed that from these questions, the concept that you will get to know will be, of course, there in the exam. Okay, so the concept understanding is important, not remembering these questions. Okay, so please focus in this session on understanding the topics. Okay, let's begin. So yeah, first question is from the animal kingdom, and the question is saying that we have to match the column 1 with column 2. Like the first characteristic here is the water vascular system, which is present in the members of family of phylum Echinodermata. Canal system is a characteristic feature of poriferans. Metamerism is found in annelids, and jointed appendages or jointed feet are present in the members of arthropoda, and hence you get your answer. Okay, so 1 ka B is present in C and D, so A and B options got deducted here. Now the canal system, D, second to D, third to C or third to A is there, so yeah, your answer will be C. Okay, so from this question you get to know that you have to remember the characteristic features or the general features also of different phylums of animal kingdom. Okay, let's see the next question. So now this question is saying that primitive animals evolved from asymmetry to defined body plan. Which phylum first showed definite radial symmetry during their life cycle? Okay, so the first member or the first phylum of animal kingdom which started to show radial symmetry was cnidarians, which are also known as coelentrates, and they includes members like hydra, like your obelia, like jellyfishes. All these animals, they are present in the phylum Cnidaria, where your first radial symmetry evolved. Also, this is the phylum where for the first time neural system evolved. Okay, let's see the next question. Now this question is from the plant morphology chapter, and it says which of the following zones of root is responsible for increase in length of the root. Increase in length is meant to grow the plant or to grow the root of the plant in a lengthwise manner or elongation. So that's why zone of elongation is the reason where lengthwise growth happens. And how it happens? It happens because of increased vacuolation inside the cells. Okay, let's see the next question. Now this is a question which involves little bit of solving. Mathematical type of question is there. A microbial culture in a lab grows in ideal conditions without any resource limitation. It means that it is going to show exponential growth. If the initial population size is 500, which means that N0 is 500, and it doubles every 2 hours, what will the population size after 8 hours? It means that if it is doubling after every 2 hours, then after 8 hours there will be 4 generations. 8 by 2 is 4. Take, we have to calculate the number after 8 hours. So just apply simply the formula n is equal to n naught 2 to the power n, where n naught is 500, 2 to the power small n is the number of generations, 4. So now 2 to the 4 to the 8 to the 16. 16 into 500 will give you how much? 16 5s are 80. So yes, it will be 8,000. Okay, it will be 8,000. Okay, it's a simple question of exponential growth curve. Okay, let's see the next question. Now here they have given you a context, they have given you some information, and on the basis of that information they are asking a question about biotic interaction. Let's see. A certain flatworm enters the host fish body through its gills and lodges in the bloodstream. It extracts nutrients over time, weakening the fish immune system and reproductive output, but not killing it outright. Which interaction best characterizes this relationship? So we know that whenever any organism it enters the host body and start to exploit that host body without killing it, then that kind of interaction is known as parasitism. If it is inside the body, if the parasite is entering inside the body of the organism, host organism, then that is known as endoparasitism. And if it attaches on the outside body just like leeches, then that is known as ectoparasitism. So this is an example of endoparasitism. B option is correct. Okay. So by solving this question, I don't mean to say that the question is going to come from parasites only, parasitism only. What I mean to say is that biotic interaction is very important topic, and question can come from that topic. It includes many interactions like commensalism, amensalism, parasitism, predation, competition, mutualism, all these things. Okay. Now, in a biodiverse tropical ecosystem, two mammalian species consume the same types of nocturnally flowering fruit. Over several decades, one population significantly decreases due to slightly slower digestive rates, affecting its foraging success. What ecological principle does this exemplify? So they are saying that there are two species, let's say A and B. They feed on the same fruit. So it means that both of them are using the same resource, but after some time, one of them started to decrease in number due to their less efficient digestive rates, and others started to increase because it is more superior in terms of their digestive rates. So this is a typical example of competitive exclusion. So competition can lead to two things: competitive exclusion, or else it can lead to competitive coexistence. But here, since the number of one of the species is decreasing and another one is increasing, that's why it is an example of competitive exclusion. Okay, and competition is a very important topic because competition can lead to evolution also. Okay, now next question from the chapter photosynthesis, one of the very important and the very hot topic for any kind of exam in which biology is asked. Okay, let's see. So this is a question from light and dark reaction, and we have to match the things. Photosystem 1, it is also known as P700 because it absorbs light of 700 nanometer. PS2, it absorbs light of 680 nanometer, that's why it is known as P680 or photosystem 680. C3 plants, their first product is a 3-carbon product which is phosphoglyceric acid. C4 plants, their first product is oxaloacetic acid. So it is one of the kind of factual question, but yes, if you will try to read the concepts regarding these all things, then you will get to know that these are very conceptually active topics. Okay? Let's see. Photosystem 1, P700. So 1 ka C. 1 ka C is present in only one option, that is B. So that's why B option is absolutely correct. Okay? Clear this thing? Let's move ahead. In C4 pathway in plants, decarboxylation happens in which part? So we know that in C4 plant, Hatch and Slack pathway happens. And in Hatch and Slack pathway, we have two cells which are involved. First cell is known as mesophyll cell, and the second cell is known as bundle sheath cell. And the dark reaction or the C3 cycle, it happens in the bundle sheath cell, in the bundle sheath cell, in the chloroplast of bundle sheath cell. Okay, so you can now see and match the option which better gets you the idea of this question. Okay, so the decarboxylation happens in the chloroplast of bundle sheath cells. What do you mean by decarboxylation? Decarboxylation is a release of CO2. We know that in order to prepare C6H12O6 or glucose, we need carbon dioxide, and that carbon dioxide is used in the C3 cycle to produce that glucose molecule. So yes, the CO2 will be released in the place where there is a cycle which is happening to produce glucose, and that cycle is known as C3 cycle, which occurs in the bundle sheath cells of your C4 plants. Okay, now next question. Which of the following statements about the plant hormone is true? So this is also one of the important topic about phytohormones or plant growth hormones. So there are total 5 hormones, plant hormones, that you have to study: auxin, gibberellin, cytokinin, ethylene, and abscisic acid. Let's see. Auxin promotes abscission. Wrong. Auxin do not promote abscission. Auxin inhibits dormancy. Auxin inhibits abscission. Okay. ABA promotes growth. No, ABA inhibits growth. Gibberellin promotes dwarfism. No, gibberellin promotes elongation or lengthwise growth of plant. Cytokinin promote nutrient mobilization. This is correct answer. Cytokinin promotes nutrient mobilization. Cytokinin promotes cytokinesis or cell division. Cytokinin is also known as anti-aging hormone. Cytokinin also promotes lateral growth of plant. Okay? So all in all, cytokinin is involved in the overall growth of the plant. So D option is absolutely correct. Okay? Let's see next question. Now, during an action potential, the role of calcium ion is 2. So action potential generation requires opening of voltage gated sodium channel. Action potential release or repolarization happens due to opening of voltage gated potassium ion channels. But traveling of action potential across 2 neurons, it happens with the release of calcium ions, because calcium ions are involved in moving or in facilitating the movement of synaptic vesicles at the end of the synaptic knob, which actually is involved in release of neurotransmitters in the synaptic cleft that actually transfers the signal to the next neuron. Okay, so calcium ions are involved in the release or in the movement of synaptic vesicles to release neurotransmitter in the presynaptic membrane. Okay, so it triggers presynaptic release of neurotransmitters. Clear? Let's move ahead. Now next question. So it's a context based question. Let's see. A research team genetically modifies a flowering plant so that a specific gene regulating differentiation of tapetum fails to express. So tapetum is a layer present in the microsporangium or the male reproductive structure of flower, which is involved in giving nutrients to the microspore for its proper maturation. Okay, so if the gene responsible for development and differentiation of tapetum is mutated, then the microspore or the pollen grains, it will not be able to mature. It will not be able to mature. So yes, let's see the option which says such things. Okay, let's see. So tapetum is essential only for providing enzymes required for endothecium thickening. No, it is not involved in endothecium thickening. It is involved in giving nutrients for the maturation of microspores. Okay? Tapetum secretes sporopollen precursors and nutritive substances. Its absence disrupts exine formation and nutrition, leading to pollen abortion and failure in anther dehiscence. This is the most appropriate answer here. And why C and D are incorrect? Because they talk about endothecium and middle layers again. So that's why they are also wrong answers. Okay, let's see the next question. Next, again, at the 8 nucleate stage of the female gametophyte in most angiosperms, which of the following accurately describes the arrangement and functional roles of the nuclei and cells involved in fertilization? We know that the female gametophyte is also known as embryo sac, and it has 7 cells and 8 nucleate condition. ठीक है, 1 2 3 4 5 6 and 7 cells are there, but nuclei are 8. 1 2 3 4 5 6 7 8 nuclei are there. And during the fertilization, this egg is fertilized and this polar nuclei is fertilized. Other cells are not fertilized at all. ठीक है, let's see which statement matches what we discussed. A option says the 8 nucleate structure consists of 8 independent cells, each capable of participating in fertilization. No, 8 independent cells are not fertilized. Only 2 cells are fertilized. The 8 nuclei are distributed into 7 cells. Correct. With the egg cell, 2 synergids and central cell containing 5 nuclei, which contribute to fertilization. Yes, it can be correct. It can be correct because the egg cell and 2 synergids and central cell contains 5 nuclei, because 2 are present here, 1 is here, and 2 nuclei are present in the central cell, which is polar nuclei, which contributes in fertilization. This can be the correct option, though we are not sure because 2 other options are also remaining. Okay, the egg cell and the 2 synergids are at the micropylar end, correct, while the 5 other nuclei are randomly distributed in the embryo sac. No, they are not randomly distributed. They are also present in their specified places. Take the 8 nucleate stage includes 7 cells, yes, with the central cell containing 2 polar nuclei. Yes, central cell contains 2 polar nuclei here, that will fuse with a sperm to form the endosperm after fertilization. This is also absolutely correct statement. Okay, so D option is the more appropriate statement in this question. Okay, clear? Let's move ahead. Now, this question says that a sterile female with rudimentary ovaries, short stature, webbed neck, broad chest is diagnosed with imbalance in copies of sex chromosome 44 plus X0. The female is diagnosed with. So, whenever there is a condition of 44 plus X0 condition, then this genotype is of a female and this female is infertile, and it is suffering, she is suffering from Turner's syndrome. Okay, and this is a question from genetics chapter, and in genetics also chromosomal disorder subtopic. Okay, and the answer here will be Turner syndrome, because this type of genotype is present in Turner syndrome, and this occurs due to monosomy of X chromosome. Monosomy of X chromosome. It occurs due to monosomy of X chromosome. Correct? Tick. Let's move ahead. A person with AB plus blood group marries with a person with B plus or B positive blood group. What are the possible blood group will be found in their children? Okay, so what are the different possibilities of blood group that can be present in their children? Let's see. So AB blood group is present only and only when the genotype looks like this, IB-IA or IA-IB. B blood group is present when the genotype is IB-IB type or IB-IO type. So from here we can get multiple possibilities. Okay, so this IB can fuse with this IB to produce IB-IB, or B blood group. This IB can fuse with this O to again produce B blood group. This A can fuse with this B to produce AB blood group. This A can fuse with this O to produce A blood group. Okay? So the total possibilities are A blood group, B blood group, B blood group and AB blood group. Okay? So there are 3 possibilities and you can see which among these possibilities are mentioned over here. O is not possible in this case. So all the options with O blood group gets cancelled. So the only remaining option is option number C, which shows the possibility of A and AB. So here we have A and AB possibilities, which is correct. Take, let's move ahead. How many non-parental combination exist when a cross between a yellow round pea plant with green wrinkled pea plant is done? Basically, they are asking about the Mendel type of dihybrid cross and they are asking about the F2 generations. So yellow and round seeds when crossed with green and wrinkled seeds, they are both dominant traits and they are also both recessive traits. Okay, round and yellow, let's denote it like this, capital Y, capital Y, capital R, capital R, and here let's denote it like this, small y, small y, small r, small r. When they will fertilize to get the F1 hybrid, it will look like this, capital Y, capital R, small r, which will all contain round and yellow seeds. But in the F2 generations, we know that it will assort independently to give you a phenotypic ratio of 9:3:1. Total 16 possibilities. 9 plus 3 plus 3 plus 1 is total 16 possibilities. Out of these 16 possibilities, this 9 and this 1 are homozygous recessive and not homozygous recessive, are actually showing both recessive and both dominant traits. So both dominant and both recessive are the parental types. So these 10, 9 plus 1, 10 will show the parental generation. But they are asking about non-parental gene combinations, which will be the 3 which are mentioned in the center. So 3 plus 3, 6. 6 by 16 is the ratio of phenotypes that will be non-parental, which matches with option number C. Okay, actually they haven't mentioned that in which generation, F2 or F1, we have to see this thing. They also haven't mentioned about do we have to see the genotype or the phenotype, but from the options we can judge that they are most probably asking about the F2 generational phenotypes. Okay, clear? So such type of questions can be asked. Such type of questions are very interesting and they can trick the students also. That's why the examiners, they generally tend to ask questions like these from the genetics chapter. Okay, let's move ahead and see the next question. Which of the following given statement about VNTR is incorrect? VNTR stands for variable number of tandem repeats. What? Variable number of tandem repeats. Variable number of tandem repeats. Okay, and these variable number of tandem repeats are present in the non-coding region of our DNA, of our genome, and they are different for every different individual except monozygotic twins. So these VNTRs, they are universal for every individual. Okay, let's see the options and judge the answer. VNTRs are located in non-coding regions of the genes. This is correct. VNTRs are identical in all individuals. No, VNTRs are identical only in monozygotic twins. Other than that, no individual shows identical VNTRs. So this is incorrect statement and we have been asked the incorrect statement with respect to VNTR. So that's why B option is incorrect, which means that it is the answer. Okay. Let's move ahead. Now next question is saying that which of the following is an incorrect statement in case of sex determination in honeybees. We know that in honeybees, drones are haploid and fertile, while worker bees and queen bees, they are diploid. ठीक है? Okay, let's see. And they have 32 chromosomes. But this drone has 16 chromosomes because it is haploid. Let's see the options. The sex. So one thing that you must note here is that they are asking about incorrect statement. Let's see. The sex determination in honeybees is based on the number of set of chromosomes. Correct. Because number of set of chromosome differs. This is correct. Next. The females are haploid having 32 chromosomes. No. Females are diploid having 32 chromosomes. This is a wrong statement and hence it is the answer because they are asking about the incorrect statements. Okay, let's move ahead. Now, so it's a factual question from the chapter of molecular basis of inheritance. I have deliberately included this question because many a times we feel that it's a conceptual unit, it's a conceptual chapter, so that's why non-conceptual things or the factual things can't be asked from this chapter, but this is a misconception. They can ask factual things and this chapter has plenty of factual things that can be asked. One of those factual things is this. Let's see. So in this question, they are basically asking about the number of bases present in different organisms. Like bacteriophage 5x174. How many base pairs it contains? It contains 5,386 base pairs. Okay. Bacteriophage lambda, it contains 48,502 base pairs. E. coli, it contains 4.6 into 10 to the power 6 base pairs. And human diploid genome, it contains 6.6 into 10 to the power 9 base pairs. Okay. So A matches with 4. A matches with 4. So B and D automatically got detected. Check. B matches with 3. B matches with 3. In A option, B is matching with 3. Other options are getting incorrect. Okay, clear? So this is the thing. So A option is correct in this question. So the motive here is that you should remember the facts from this chapter also. Okay, let's move ahead. The promoter and the terminator sequence in a gene are located towards which of the following? So we know that whenever we have to undergo transcription, we talk about cistron. And this cistron, it has some regions. The initial point of this cistron is called as promoter. Middle one is known as structural gene and the ending is known as terminator. This is 3' and this is 5' end. This is 5' and this is 3' end. We actually always read the cistron with respect to the coding strand. So there are two strands. One is known as coding strand and one is known as a template strand. So template strand is a strand whose information are read by the RNA polymerase and the complementary RNA is made. But the coding strand is that strand whose bases are exactly the same as that of the mRNA, except that the DNA contains thymine and the RNA contains uracil. So we actually read all the information with respect to the coding strand. So promoter is located at the 5' end of the coding strand and terminator is located at the 3' end of the coding strand. So what could be the answer here? So 5' end and 3' end of the coding strand. A option is correct. Okay, promoter is located at the 5' end of the coding strand. Terminator is located at the 3' end of the coding strand. Okay, clear? Let's move ahead. So you can also say that bhaiya, this option is also correct. But like I said, that we always give or we always study a cistron with respect to the coding strand. That's why if such kind of question which seems to be ambiguous to you, in that question the most appropriate answer will be the correct answer. Okay, Let's move ahead. So you must focus on getting to the most appropriate answer. Clear? Let's move ahead. So this is very important topic from which question has been asked. It's about Hardy-Weinberg principle. And in Hardy-Weinberg principle, we know that we have allelic frequencies P plus Q is equal to 1, whose sum is equal to 1. And we have genotypic frequencies P square plus 2PQ plus Q square is also equal to 1. But these two cases only apply if and only if Hardy-Weinberg principle is being followed. But in this question, there is no such thing which is written. Let's see this question. It is saying that suppose there is a population of 1,000 individuals in an island, of which genotype A found is 270. A genotype is found in 270 individuals out of these 1,000. A small a is found in 590 individuals and small a is found in 140 individuals. Now they are saying that what is the frequency of capital A? What is the frequency of this capital A in this population? Let's see. We know that there are 1,000 individuals, so the total allele in this population will be double of this thing. So 1,000 into 2. Okay? Total allele will be 1,000 into 2, which is 2,000. Because one individual contains two alleles of one trait, like capital A. 270 individuals contains capital A genotype, which means how many alleles? Double this 270. Okay. Now they are asking about the allelic frequency of capital A. Let's see. Capital A here, 270. 270 into 2 capital A are present here, and here 590 capital A are present here. 590 capital A are present here, and if we divide it by 2000, we'll get the allelic frequency of capital A, that we'll call small p. P for capital A. Okay? How much it will come out to be? 590 plus 270 to 270 to 2, how much will it be? 400, 470, 540. 540 by 2000, which will come out to be 1090, 1130 or something. Approximately, we are getting the approximate data here. It is approximately near to 0.5. So you can just see the option and judge the answer. Which option is somewhere near to 0.5? This one, है ना? So this one is the answer, 0.5. Clear? Clear? Okay. Let's move ahead. Now this question, it seems to be a long question, but it is actually a very simple question, because it is just asking about the type of selections. If you know the type of selection and how it behaves, you will get to know about the answers here. Let's see. So you must be focusing on reading these statements, and on the basis of that, just matches with the column number 1. First question, it says that in a century, trees were planted with 2 types of seeds. Okay, one big and one small. Birds with large beak could crack open bigger seeds. So large beaks are there, while the smaller beaks could crack smaller seeds. Okay, smaller beaks are also there. Okay, now later birds with average beak size decreased. So large beak sized birds got selected, small beak sized bird got selected, but the average beak sized bird, they got deselected. But deselected is not a correct word, but they got diminished in this population. So it's a simple example of disruptive selection, because 2 extremes are getting selected. Okay? So it's a simple example of disruptive selection. So just match column number 1, this statement number 1 with B. So B ka 1 is present in A, B and D. So C option is gone here. Okay, now let's see. Upon prescribed antibiotics, a person did not complete her course and stopped midway. A few resistant bacteria survived and multiplied, causing the disease again. ठीक है, it can be an example of directional selection. It can be an example of directional selection, but it can also be an example of stabilizing selection. So that's why in such cases, let's move on to the next statement and confirm this thing. Let's see. According to hospital data, most mortality occurred with babies with very high and very low birth weights. Only babies with normal weight have a high survival chance. It means that the average size weight baby, they have a higher chance of getting selected or surviving. So it's an example of stabilizing selection. It's an example of stabilizing selection. ठीक, तो third matches with C. Third matches with C. So only in A option, third is matching with C. Now let's see fourth option also. A study was conducted in which humans were kept in extremely low and extremely high regions. They found that in low temperatures, human developed symptoms of hypothermia, while in high temperatures, it resulted in heat strokes. Only humans in normal temperature were healthy. So it means that fourth is also matching with stabilizing selection. Fourth is also matching with stabilizing selection. So 3 and 4 both are matching with stabilizing selection, and which is a case in A. ठीक है, A option is matching with all these statements. Okay, so A is the answer here. Basically, the point here is that you must understand what is directional, disruptive, and stabilizing selection. Other than that, if you know all these three terms, then you will be able to get an idea of which kind of selection is taking place in any kind of example, not only in these four, in any kind of example. So just understand these three terms. Other than that, you will be able to get it. Okay, let's see next question. Marsupials in Australia and placentals in mammals and placental mammals in other continents have evolved to fill similar ecological roles. This illustrates. Actually, when we compare placentals with marsupials, we know that we are comparing two different groups of animals, marsupials and placentals. But it is seen that both of them, they have similar kind of body structure, they have evolved similar kind of functional roles in their own ecological niches. But their ecological niches, they are very similar. That's why they have evolved such kind of similar structure and function. But still, they are of different origins. So different origin but same function denotes convergent evolution or analogy. So that's why it matches with option number C. Tick. Let's move ahead. Now, this question again is saying a population of beetles showed a shift in color from light green to dark green due to bird predation. This is an example of. So when one extreme shifts to another extreme, then that is moving in one type of direction, one direction. है ना? So in that case, we say that directional selection has taken place, or selection which is acting is moving in one direction. So that's why this is an example of directional selection. Clear? Okay, let's move ahead. Now this is a simple example or simple question of Hardy-Weinberg principle. In the previous questions, it was not mentioned that the population is following Hardy-Weinberg equilibrium, but in this population, in this question, Hardy-Weinberg equilibrium is being followed. So that's why we can apply these two formula here. P plus Q is equal to 1 P square plus 2 PQ plus Q square is equal to 1. These two formula can be applied here in this case because it is following Hardy-Weinberg equilibrium. Let's see what the question is saying. In a population, the frequency of allele capital A is 0.6. Okay. What is the frequency of heterozygous genotype capital A small a? Heterozygous genotype is denoted by 2pq. Okay. So capital A small a is denoted by 2pq. Okay. It is denoted by 2pq. P is given to us, and we know that if P is there, we can of course get Q, because Q is 1 minus P. So 1 minus 0.6 is 0.4. So you have got Q, you have P from the question. So you can calculate P, 2pq, 0.6 into 0.4, and 6 for the 24. 6 for the 24, 2 is a 48. 0.48 is the answer. Yes, 0.48 is the answer here. Okay, simple, very easy question. But students generally panic when they see question from Hardy-Weinberg equilibrium, and they end up losing marks from this question, which is a very important topic and very easy topic also. Check, let's see. According to the 10% law of energy transfer, if 10,000 kilocalorie of energy is available at the producer level, if 10,000 kilocalorie energy is present at the producer level, then how much energy will be present at the secondary consumer level? So producer, then primary consumer, and then secondary consumer. Okay, 10,000 at producer. From 10% law, we know that at every trophic level, 10% energy is transferred of the previous trophic level. So the energy present here, only 10% of this will be transferred to the next trophic level. So 10,000 if present in producer, 1,000 will be in primary consumer, and 100 will be present in secondary consumer. So 100 kilocalorie will be present in secondary consumer. So that's why B will be the answer here. Okay, clear? Let's see. Which of the following is not part of Abel quartet that leads to biodiversity loss? We know that biodiversity loss, it happens due to 4 main reasons. Those are known as Abel quartet. And what are those 4 reasons? These 4 reasons are habitat loss and fragmentation, overexploitation, alien species invasion, and co-extinction. Other than that, if there is any other option in this question, that will be a wrong answer. Okay, that will not be an example of Abel quartet. Let's see. Habitat loss, okay. Overexploitation, introduction of alien species, increased biodiversity hotspots. This is not amongst the Abel quartet. Okay. In spite of this, co-extinction could have been written, should have been written. Okay. Now next question. In a population experiencing logistic growth, the growth rate is highest when the population size is. In logistic growth curve, we know that the formula looks like this. dn upon dt is equal to Rn k minus n upon k, where this k represents carrying capacity. This k represents carrying capacity. This k represents carrying capacity. In logistic growth, when the population or when the number of individuals reaches half of carrying capacity, at that time the growth rate is maximum. Okay, at that time growth rate is maximum. Okay, even if we plot a graph here with the rate of growth with the number of individual, it will look like this. And here the maximum rate of growth will be achieved at k by 2. Okay, so the answer will be C. Okay, clear? Let's move ahead. Consider a population where the initial size of the population is 1000, N0 is 1000. Okay. During a certain time period, 200 individuals are born and 50 die. Okay? It means that how many total individuals are added? 200 minus 50, which will be 150. Okay? And at the same time, 100 individuals immigrate into the population and 150 emigrate. How many? What will be the change due to migration? 100 minus 150, which will be minus 50. Okay. What will be the population size at the end of this period? Initial size was this, 150 is added, 50 is lost. So 1000 plus 150 minus 50, which will be 1100, which will be 1100. So 1100 will be the answer here. Okay? I know that D option also says 1100, but that can be ignored. That is a that is some technical glitch. Okay? Any kind of thing could have been written here, but yes, the correct answer is 1100. Okay, clear? Let's move ahead. Now, a population of microorganisms was introduced into a nutrient-rich medium. Initially, the population doubled every year. Okay, initially the population doubled every year. After a certain point, although reproduction continued, the rate of growth slowed and eventually plateaued. Which of the following best explains this phenomena? We know that from this question that this population of bacteria, they actually are showing logistic growth, because initially they are increasing, but after that they are plateauing. Okay? If we plot a graph number of individuals with time, it will look like this. Okay, it will look like this. But in logistic growth curve, why after some time the plateau occurs? Why the growth ceases to happen? Because the resources in any population are limited. So it is because the resources are limited and the population starts to grow, starts to rise in an uncontrolled manner initially. So that's why the resources become limited and it reaches a plateau. Any option like that? Let's see the option. Yes, C option. Resource limitation caused the population to stabilize after initial rapid growth. Clear? Clear this thing? Okay, let's move ahead. Now, which mechanism of acquired immunity involves the production of antibodies? So, antibody production is mediated by humoral immunity. So, there are two types of immunity here. Immunity is divided into two parts. First one is known as innate immunity and the second one is known as acquired immunity. In acquired immunity, we have two types of immunity. One is cell-mediated immunity and one is humoral immunity. Okay? Cell-mediated immunity is mediated by T lymphocytes. Humoral immunity is mediated by B lymphocytes. And we know that B lymphocytes, they are involved in production of antibodies. But T cells, they are involved in helping the production of antibodies. So that's why antibody production happens due to B cells, which is an example of humoral immunity. So B option is correct. Okay? Clear this thing? Let's move ahead. Cancer involves uncontrolled cell division. We know that tumor or a cancer, it involves uncontrolled cell division leading to formation of a lump in any part of our body. Okay. Mutation in which type of gene is most directly responsible for transforming a normal cell into a cancerous cell? So a normal cell, how it enters into the cancerous cell or neoplastic growth cycle? A normal cell, its cell division is regulated, but a cancerous cell, its cell division is not regulated at all. So which are the genes that are involved in the regulation of cell division of normal cells? They are basically two types of genes. One is tumor suppressor gene and the second one is proto-oncogene. Okay? So other than that, also there are many other genes, but yes, this tumor suppressor gene, these actually negatively suppress or negatively actually does some things which actually inhibits cell division. Okay, but if these tumor suppressor genes, they are mutated, then that regulation or that inhibition will not happen. So that's why if something is not inhibited from division, then they will of course start to divide in an uncontrolled manner. So that's why tumor suppressor gene, mutation in tumor suppressor gene, or loss of function mutation in tumor suppressor gene, they actually leads to uncontrolled cell division. Okay, now let's see the next question. Vaccination. What is a vaccination? Vaccination is an example of active immunity. Vaccination provides protection by training the immune system to recognize and combat pathogens. Which of the following best describes how vaccination leads to immunity? Actually, in vaccination is given to a person who is not diseased by that disease against which vaccine is being added here. Okay? Now, in that vaccine, we don't add any kind of medicine or drug. We only give that virus or that pathogen or that bacteria, that protozoan, to the body of a person in which we are trying to mediate that active immunity process. Okay? So we actually give the weakened or attenuated pathogen to the person, so that the immune system of that person, it recognizes this pathogen which has been ingested in his body, in his or her body, and it starts to produce memory against that infection. So it is that memory which is present in the immune cells which is showed in the next subsequent infections. In the next subsequent infection, the body's immune system, it knows that what kind of pathogen is it and what kind of responses should it generate to kill that pathogen. So it is basically due to memory produced by B and T lymphocytes. Any option? Yes, C option. So it induces, vaccination induces memory B and T lymphocytes formation. Okay, clear. Now let's move ahead. Again, autoimmune disease occurs when the immune system attacks the body's own tissue. There are some disorders in which our body tissues or our body's immune system, it attacks our body's own cells, okay, by mistake. They are asking simply about the examples of those autoimmune disorders. So yes, osteoarthritis is not an example of autoimmune disease. It's a hormonal disorder in which bone density is decreased. Rheumatoid arthritis is an example of autoimmune disorders. Likewise, myasthenia gravis, Hashimoto's disease, chronic hepatitis, these all are also an example of autoimmune disorder. Alzheimer's disease is mainly neurodegenerative disorder. Diabetes, type 2 diabetes is actually related to lack of insulin production, but type 1 diabetes can be an example of autoimmune disorder. Okay, check. So you should remember all those examples. Now next, as AIDS progresses, patients suffer from infections they could normally resist. The opportunistic infection commonly associated with advanced AIDS is. From this question, what I mean to say is that you should remember all the things regarding AIDS. So AIDS is a disorder which is known as acquired immunodeficiency disorder. And once the immune system is suppressed, then we tend to get any kind of infection because the immunity is low. Okay, and the most common type of opportunistic infection that occurs in case of AIDS is tuberculosis, TB, which is a bacterial infection caused by Mycobacterium tuberculosis. Okay, clear? Let's move ahead and see the next question. Now this question is from the chapter of Biotechnology and it's a Mazda type question. It says origin of replication. What is origin of replication? Origin of replication is a site from where replication originates. Okay. Yes, C option. Sequence from where replication starts. So A matches with C. Selectable markers, there are some sequences or there are some genes in the plasmid DNA which actually is involved in selecting transformant and deselecting non-transformants. Okay, any option like that? Yes, B. Helps in identifying and eliminating non-transformants and selectively permitting the growth of transformants. Cloning sites are the sites in which our gene of interest is inserted. A option. And restriction endonuclease, it actually are naturally produced in the bacteria as a defense system against bacteriophages. So 1 ka 3, 2 ka B. 2 ka B is only present in C option. So C is the correct answer here. Okay. Let's see the next question. The Bt toxin is not toxic to human beings because why Bt toxin is not toxic to humans? First of all, let me tell you, Bt toxin is actually a protein known as cryoprotein, and this cryoprotein, it is produced initially in an inactive manner and it is activated once it gets alkaline pH. So this is the reason why it is inactive in humans, because it needs alkaline pH to get activated, and that alkaline pH it gets inside the gut of any insect. Any option like that? Ha. So Bt toxin, it do not infect or do not affect human because it needs conversion of pro-Bt toxin to Bt toxin only takes place in highly alkaline condition, which is D option, and that's why D option is correct. Clear? Let's see. Now this question, it's saying that genomic DNA is digested with ALU1 restriction enzyme which has 4 base. Let's say those bases are ATGC. Okay. Now they are asking that what is the frequency with which it will cut the DNA assuming a random distribution of bases in its genome. In such cases, we have to consider all the possibilities. Okay. If it's a 4 base cutter, if this enzyme, this restriction enzyme is a 4 base cutter, then it can cut at AAA or TTTT or GGGG or CCCC, any kind of base could be there, but it's a specific kind of but restriction enzymes are specific kind of cutters. Okay, so they cut at very specific base position, and that specific base position, let's say it is ATGC. So what is the chance that it will get ATGC as a sequence? The chance here is 4 to the power 4, because we have 4 bases and it is also a 4 base cutter. So that's why 4 to the power 4, and 4 to the power 4 will come out to be 256. So it will cut once in 256 bases. This is a probability. Okay, so it will cut once in every 256 bases. Okay, answer is C here. Clear? Let's move ahead and see the next question. Now the next question, what is it saying? Which of the following properties is helpful for selecting transformants from non-transformant colonies in bacteria? Simple question of selectable markers. Simple question of selectable markers. Simple question of selectable marker. What is it saying? It is saying basically, or the selectable marker, what is it? They are actually the gene sequences which selects the bacteria which contains a plasmid having our gene of interest in it, and deselecting those plasmid or those bacteria containing plasmids in which our gene of interest hasn't been inserted. Okay? And these selectable markers are mainly antibiotic resistance genes or beta galactosidase genes, such kind of things. Take. So let's see the options. Presence of more than one recognition site in the vector DNA? No, this is a very bad thing. If in a plasmid more than one recognition site is present, then we face difficulty in selecting it. Okay. Insertion of alien DNA into vector DNA leads to insertional activation. Insertional activation of the marker. Insertional activation of the marker. It's a wrong statement. It's insertional inactivation. It's insertional inactivation of the marker. Okay, so B option is incorrect again. The antibiotic resistance gene gets inactivated due to the insertion of alien DNA. This is absolutely correct statement. This is absolutely correct statement. So C is the correct answer here. Okay, let's move ahead. Now this question is saying which one of these DNA molecules will have the highest melting temperature? The highest melting temperature will be present in the DNA which has highest GC content. Why? Because the G and C, these GC, they have 3 hydrogen bonds in them, and A and T, they have only 2 hydrogen bonds between them. So the bases which contains, or the DNA which will contain more amount of hydrogen bond, their melting temperature will be higher because it will be much difficult to break them as compared to AT rich DNA. So that's why the GC rich DNA will have higher chance of getting, or higher chance of having more melting point. Okay? But yes, melting point also depends on the conformational or the structural manner in which DNA is present, but mainly it depends on the GC or AT content. Take the one which contains higher GC content is going to have more melting point. Take. Now, this is your PYQ of IAT. It is saying, or it is from the topic of polymerase chain reaction, and it is saying that polymerase chain reaction is used to amplify a gene of interest. If after 30 cycles we get 1 billion DNA, which means 10 to the power 9 base pairs, approximately how many copies of gene of interest will be present at the end of 20th cycle? After 20 cycles, how many DNA will be present? Okay, so they have given information about 30 cycles. They are saying that after 30 cycles, we are going to get 10 to the power 9 bases. How many DNA will be present? Were present after 20 cycles? Simple question, needs simple formula. So yes, just apply the concept of n is equal to n naught 2 to the power n. So n after 30 cycles are how much? 10 to the power 9, n naught upon 2 to the power 30. Okay? So n naught, or the initial number of DNA, was 10 to the power 9 upon 2 to the power 30. Check. Now just apply this n naught to the 20 cycle formula. Take again, n after 20 cycle will be n naught, which we have calculated, 10 to the power 9 upon 2 to the power 30 into 2 to the power 20, because they are asking about 20 cycle. Take, just calculate it and you will get the answer. 2 to the power 30 minus 2 to the power 20 will be 2 to the power 10, and we know that 2 to the power 10 comes out to be approximately 1024, which is approximately 1000. Let's write it like this, 10 to the power 3, and if we cut it, it will come out to be 10 to the power 6, which is 1 million DNA. Okay, which is 1 million DNA. Clear? So any topic can be molded in any manner. Okay? You should be clear with your concepts and how to apply those concepts. Okay? And if this is your approach, then you will, of course, you will be able to solve all the questions of biology in the actual exam. Okay? Let's see. Now they are saying Chlamydomonas and Chlorella, earlier placed in algal groups, were brought together in which correct group? According to R.H. Whittaker's system of classification, all the unicellular eukaryotes are placed under the kingdom Protista, and these are unicellular eukaryotes. That's why, though they looks like algae, they behave like algae, but since they are unicellular, that's why they are put under this kingdom known as kingdom Protista. Clear? Let's see the next question. Once the perfect stage of this fungus are discovered, they are often moved to the other classes of fungi. This statement is true for which of the following class of fungi? We know that there is a group of fungi or the phylum of fungi which do not contain any sexual stage in their life, in their whole life. But if any member or any organism, any fungus of that phylum starts to possess a sexual cycle, then they are placed or they are taken up from that group and they are placed in some other group. And what is that group? That group is Deuteromycetes. So the members of phylum Deuteromycetes, or the fungus present in Deuteromycete phylum of kingdom fungi, they do not have any sexual stage. If sexual stage is discovered in any member of that group, they are simply picked up and they are put in some other phylums of fungi. Clear? Okay. Now let's move ahead to the next question. What is it saying? Let's see. It's saying match columns. Match the columns. So mixotrophic nutrition. What do you mean by mixotrophic nutrition? Mixotrophic nutrition basically means both kind of nutrition, autotrophic and heterotrophic nutrition. If any organism contains both autotrophic and heterotrophic nutrition in them, they are said that they are possessing mixotrophic nutrition, and it is present in euglena. Red tides are shown by dinoflagellates goniolax. Free RNA molecules without protein coats are actually viroids. Obligate anaerobes are methanogens, which are archaea bacteria. Okay? So 1 to 3 or P to 3 is present in only C option. You could have got the answer by just matching P. Okay? Because other options are not matching. Okay? Clear? Let's move ahead. Select the incorrect pair or incorrect statement. This is a question from plant kingdom. Let's see. Option number A. It's saying that in bryophytes, the dominant phase is gametophyte. Correct. In pteridophytes, the dominant phase is gametophyte. This is incorrect. In pteridophytes, though the life cycle is haplodiplontic, but the dominant phase is sporophytic. So that's why this is an incorrect statement, and they were asking about incorrect statement. Okay? Clear? Other statements, let's see other statements also. Some algae show a haplodiplontic life cycle. Correct. Both bryophytes and pteridophytes are of haplodiplontic life cycle. This is also correct. Take? Clear? Let's move ahead. Now, which or what is a phenomenon of chromosomes doubling during cell division? By what means is it induced and in which phase? The doubling of chromosome is known as polyploidy. Okay? Not doubling, tripling or quadrupling is also known as polyploidy. Okay? So polyteny and polydipsia is gone. Okay? So among B and D, anyone could be the answer. By what means is it induced? So it is induced by a chemical drug known as colchicine, which actually is involved in inhibiting spindle fiber formation in the cell division process, and it affects mainly metaphase of cell division. Yes, so D option is correct. So polyploidy, colchicine and metaphase. Clear? Clear? Let's see the next question. How does colchicine inhibit the cell cycle? So I already told you this thing in the last question, that colchicine, it is involved in inhibiting spindle fiber formation or spindle microtubular arrangement in spindle fiber formation. That's how it inhibits cell division, or it induces polyploidy. What could be the answer here? It interrupts in the formation of spindle fiber. Clear? Let's see the next question. Now, this type of question are very important because they are saying, or they are trying to ask about the chromosome number in different phases of cell division. And they are asking about human cells. So human ovum, human ovum, they contains haploid set of chromosomes. So it is 23. So D ka 3. D ka 3, D ka 3, D ka 3. A option is gone. ठीक. Spermatogonia pre-replication, it will contain 46 chromosomes. Because pre-replication 46 chromosomes and 46 chromatids are present. So C ka 1. C ka 1 is given only in option B. Let's see the other option also. G1 phase also 46 chromosomes. B ka 1. And G2 phase contains 92 chromatids. Because in G2 phase or in S phase, the replication happens, which gives rise to one more set of DNA. That is also considered as a separate chromatid. Okay. So option B is the absolutely correct answer. Take. Next question. This question is saying that given below are 4 schematics that describe the dependence of the rate of an enzymatic reaction on temperature. Means that they have given 4 graphs and they are saying that this is a graph of dependence of enzyme activity with change in temperature. Okay, and they are saying that they are comparing thermophilic and psychrophilic organisms. So thermophilic organisms, they grow at higher temperatures, and psychrophilic organisms, they grow at lower temperatures. Okay. Now they are saying that which among these 4 graphs PQRS coincides with thermophilic and psychrophilic organisms. So let me tell you that the dependence of the dependence of temperature on enzyme activity, or the dependence of enzyme activity with temperature, it looks like this. Why? Because at any optimum temperature, the enzyme activity is highest. Okay? And below that temperature and above that temperature, the enzyme activity is lowered. So there is one optimum temperature of any enzyme. It can be of psychrophilic and thermophilic organism, any one among them. All of them works best at their own optimum temperature. Their optimum temperature could vary. It can be 100 degrees Celsius, it could be 37 degrees Celsius as in our body. It can be 0 degrees Celsius also. But they will work best at their optimum temperature. And hence the graph will look like this always. It can shift here, it can shift here, but the shape will look like this in all the cases. So for both thermophilic and psychrophilic organisms, R will be the correct option. Okay, so both are R's. Okay, C option is the absolutely correct answer. Clear? Clear? Let's move ahead. Now, inulin is a polymer of fructose. Inulin is a polymer of fructose, and chitin is a polymer of N-acetylglucosamine. N-acetylglucosamine. Now you can just compare the answers. Fructose and glucosamine. Fructose and glucosamine is the correct answer. So the motive here to give these questions is to make sure that you consider biomolecules as an important chapter. It's an important chapter. Specifically, enzyme activity is a very important topic, and also other parts are also very important. But yes, biomolecule as a whole, this chapter is important with respect to any kind of exam in which biology is asked. Okay, let's move on to the last question. It's saying that which term describes the reduced fitness of a population due to mating of closely related individuals. So like if I give you an example here, whenever 2 closely related individuals are mated for multiple generations, then the harmful characteristics that persist in their family, it starts to rise. Okay? Its occurrence starts to rise. So that's why close mating is prohibited in many countries, including India. And hence, 2 two closely related individuals are never mated in any animals. ठीक है? So and those harmful traits that are expressed in closely related in the after the mating of closely related individuals, those harmful traits are known as inbreeding depressions. Those harmful traits are known as inbreeding depression. Okay, so these were the 50 important questions that I wanted to discuss with you. Why? Because they are not those questions which are going to come in your exam, but they are those questions which actually caters the important topics with respect to your exams. Okay, I hope these questions will help you in understanding the concepts underlying these questions, and also they will help you in enhancing your marks in the exam, be it be a PCB student or it's a PCM students. And I hope that you do your best in the exam and you actually get selected in your dream institutes. But for that you have to consistently keep doing hard work, and yes, hard work always pays. So yes, keep doing your part of hard work, and rest of all, please let it be, है ना? And whatever will happen, it will be good, it will be best for you. So what I can do is to give you a valuable content, and that I am doing. Okay, so best of luck to you, and only 10 days are remaining, so keep doing your part of hard work. Okay, bye bye.